So I'm going to talk about some of my work on wheat. So I do work a lot on rice. So wheat's on Thales side, rice is in the middle, and then also on corn. So it's kind of a perfect background for what I do. So the title of my talk is that wild wheat can be used as a source for enhancing drought adaptation in wheat, which I call bread wheat or the wheat that we eat. So my lab is interested in how water affects crop productivity. Today, the key factors that I focus on are water quantity, and those would be drought and flooding. So we take a look at the effects of these two stressors on crop productivity. We also work on water quality, so a lot on salinity, especially in rice, and more recently in heavy metals, because they do affect the productivity as well as food safety. The idea or the track that we take is to combine the physiology at whole plant level with the genetics to understand what makes some of these crops drought and salt tolerant. And the emphasis, as I said, is on cereals. And the reason for that is that if you just look at rice and wheat and the number of calories that humans consume, combined, these two crops basically account for nearly 50% of all the calories that humans take in every year for their survival. So that kind of puts a lot of value in any discovery that we can make in terms of how it would impact food security. So the linkage of cereals and irrigation is quite interesting. As most of you are well aware, that we use irrigation in about 20% of our land that we cultivate, but that 20% of that land produces about 40% of the food. So that kind of is like a clear impact of what water can do for food production. But what's striking for me when I learned this many years ago was that about 60% of all that irrigation water is being used for cereal production, and that too like a handful of major cereals that we consume globally. So that does pose a challenge because our freshwater supplies are limited, and there's constant pressure because of urbanization and other uses on how much is available. So if you have unpredictability in the precipitation events, that could really change the game regionally, sometimes much larger continentally for how much grains can be produced with the given fresh water. So how a plant interacts with water, we've heard yesterday about the vapor pressure deficit, but there's a very, very direct interaction between the plant and the amount of water that's available, and that's through roots. So today I'm going to focus a fair bit on some of the work we've done collaboratively on roots. So the first question is, why roots? Obviously, there's a direct link. Roots are the conduit for taking up most of the water, if almost all of the water that the plants are going to take in. But when we think of plant productivity, one of the major events was Green Revolution. So I'm showing you some wheat lines, and the gene that they were tinkering with get us the semi-dwarf varieties, and we've seen this in wheat and rice, also was decreasing not only the height of the plant above ground, but it was also altering and reducing the length of the roots and the architecture of the roots. So when people have compared what the breeding efforts from Green Revolution have done to the roots with the varieties that were being grown or land races that were being grown before that, there's a striking difference in how intricate and how deep the roots go. So we want to understand that a little bit better. So this is a picture that I really like to share. Next year it would be 100 years for this picture, or the drawing done by John Weaver, a professor at UNL. where he looked at one particular land race, and he looked at in the dry land situation and also in the irrigated situation. So the same genetics, but the response that you see to these, the conditions, water availability, to be more specific, is quite dramatic. So the question we want to ask is, so this is the environment, as Sala mentioned, the E side of it, and the G is a constant. So what if we actually explore if we expand the scope of G, which is the genetics or the genotype. I will use the word genotype, which basically means the genetic makeup of that particular individual or plant in the entirety. So what if we actually try to expand that range of G and then see how it interacts with E? In this case, it would be whether it's irrigated or whether it's dry land. So that's kind of like the central idea that I want to discuss or share with you. So that brings us to why I do that. The simple explanation is that on our so it'd be right side, on your right side, you have those uniform-looking plants in green, and that's kind of what you're seeing in all these three panels. It's like highly developed, adapted to that particular environment, but also under great selection pressure. So what that means is that as humans, through domestication and then through development through plant breeding, and then developing even modern cultivars with all the tools that some of them were shared in the lunch presentation with you yesterday. So as we keep selecting, we are decreasing the genetic diversity that we take forward for our future crops. But that would have been fine because we were fine-tuning it, but the challenge is that our E is changing all the time, but the G is becoming too narrow. So if you have that situation, so the idea that I'm going to present is not new. I mean, we've done that for many decades, is can we take one of those wild species and using all the tools that we have, can we expand the G and see what those roots look like? And we take the example of roots, but it could be, grain size, it could be protein content, it could be starch digestibility, any trait that is desirable. So that's the general idea. So essentially, what I'm going to show you in a kindergarten version would be there's a whole bunch of jelly beans. And I want to try and see if we can take a few extra colors and add those in play and see what happens. So wheat's a hexaploid. What that means is it has 3 different genomes all packed in the same nucleus inside every cell. They're distinct. They operate like the other one doesn't exist to some degree. And so it's all started with hybridization in the wild, and the example I'm going to use here is of a durum wheat, which is shown here on the bottom, and it has 2 genomes instead of 3, and just above it is the wild relative of that Triticum durum. So the difference between the wheat that you see in this picture and on the right side is the bred wheat, and the one on the left at the bottom is the durum wheat, which is what you use for pasta. So that's, I know wheat domestication could be a course. That's the amount of knowledge that we have, but I just wanted to lay it up so that you have context. So the wild relative of durum wheat that we selected is called Wild Emmer Wheat. It's been known for more than 100 years to have genes that I mentioned, right? Those extra colors of jelly beans that could confer drought tolerance, salt tolerance. It could also improve the nutritional density of the grains through like grain minerals and protein content. So this is what it looks like in the wild. You would see a whole bunch of these white, black or dark purple spikes in a otherwise oat patch in the wild. So that's the wheat that we're going to talk about. The reason we picked this up is because a few years ago, a whole genome was sequenced. So that is very important for the studies that we want to do, because in the end, we want see if new genes have been brought in, and we want to have that, be able to have the sequence information so we can actually know that we brought some new genes in. So I won't labor on the point. I guess every time I move my arm, it does it. So I probably so I won't labor on the genetics. So this was done by our collaborators, where they went through 8 generations of combining elite European durum wheat. So if you've had Barilla pasta, essentially you've had these cultivar called Suevo with Zavitan, which was the wild emmer that was sequenced a few years ago. And they combined them to let the DNA intermix through classical breeding. And then at each generation, they allowed the plants to grow in the field. Anything that was too tall and would fall over, or anything that was too short is not going to make up for good yield, were dropped out. So in the end, they had about 50 lines that had a mixture of DNA, and I'll show you what it looks like in an Excel sheet. So, but what they did also was they then followed it up with back crossing it, which is basically making it more similar to the Barilla pasta or Suevo, and so that we still have those traits fixing more strongly, so we don't see a lot of noise genetically when we do these experiments. So so this is what those roughly about 50 lines look like. On your left, the extreme left column is the blue, which is the wild wheat, and on our extreme right is Suevo, which is the elite cultivar. What it is showing is through genetic markers the mosaic that we have for all the chromosomes, which are also listed like as 1A, 1B, and so on, because I mentioned that you have A and B genomes. So since it's a diploid state, so we have 2 copies of each. So what it shows is that we have a fairly good sprinkling of the wild genome or wild DNA in an otherwise largely red, very similar to the elite line. So this was the material that we started working with. So what I'm describing to you now is what the experiments that we did at Nebraska using one of the facility that we have across the street, the high throughput imaging system for phenomics. So this is so what we did was essentially we grow these plants, and then the system allows us to impose very finely tuned water stress. So what that allows us to do is that we can measure every day how much water a plant's using, and then we can replace that amount of water. But the cool thing is the same plant also gets imaged in multiple phone booths with different cameras from the top and the side, and we took those images. So when we combine these two, you could get a very good sense of how much water is being used and how much shoot growth is taking place on a daily basis. So when you're thinking about water use efficiency, classically, we would think of a much longer window, but we're using this digital and more precise weighing measures and measuring. We can actually do that, on a daily basis. So just to kind of show you that what we call projected shoot area, which is basically above ground parts of the plant that you capture by counting the pixels from multiple angles. It has a very, very high correlation with shoot fresh weight or dry weight. In short, what we're saying is that taking an image and using our software is a very accurate way of picking how big the plant is going above ground. We use this, there's more detail than I want to talk about, but basically we used a whole bunch of parameters like how tall it is, how wide it is, what does it do, how much water does it use every day. So for all those 50 lines that we call like nils, because they're nearly isogenic lines, we can have all those parameters. And this is what the plants look like. So the diversity that we have generated from that crossing of the wild to the Suevo and then bringing back the nils that look like reasonably good for field look like this. So if you look at the top one, you have the well-watered, and if you look at the bottom one, it's water limited. So each pair, top and bottom, coincide. The parent on the left is the Barilla pasta Suevo. So you can see how some plants are not showing as much of a response, whereas like the number 2 has a huge change from shoot growth, to not as much shoot growth in the bottom half. So this was kind of done for up to, these plants are 51 days old. So we then kind of did something which is kind of easier to look at a lot of data together. We made a heat map of all the digital traits that we had and then clustered them, which means we grouped them together based on their response. So we had lines that were highly stable, which didn't change much, lines that were highly plastic that changed quite a bit, and then there were some small plants and there were some really susceptible plants. So the elite cultivar, Suevo, kind of went to the extreme right and was labeled as very drought susceptible. So the red color here is like a lot more value, higher value, and blue is lower values. So we selected a handful of genotypes that we thought would be really interesting from this. And those were kind of focused on these high stability and high plasticity, because we wanted to know, what is really tolerant by not responding and what's really plastic by responding very quickly. And so this is an example of some of the lines that we selected. Suevo obviously is drought susceptible. The dark blue line is well watered, and the turquoise is water limited. So as you look at the shoot growth, and your x-axis is the count days, you see a fairly strong drop. Then you have this particular nil called 2, that had very high plasticity. So essentially, it was growing much bigger. That was the second one on the row that I showed. And then it also responded very dramatically to water limitation. And the highly stable ones only started to show differences in their shoot growth much later in terms of the day count. So those were kind of our more stable nils. So we also looked at how the photosynthesis, the whole plant, the classical things you would have done with these plants in like 20 years ago. So because they're still valuable, so we looked at how much carbon they were capturing, and so these, it was clear that the stable lines were not making much of a difference in carbon capture. They were really tolerant to the stress that we had imposed. We then did something, a little bit more laborious. We washed all the roots for these 50-some plants. So I'm showing you the ones that we had selected to kind of show what the root architecture looks like. So what caught our eye very early on was that the Suevo, which is the parent, the drought-sensitive parent, it has somewhat of a similar, maybe a little bit better root system. But the second line, the NIL 20-2, which had this huge plasticity, so it was going from a very big shoot architecture, growth. It switched to putting most of its carbon into the roots. So that's the kind of trait that we wanted to understand, what is it that drives, this change in a decision by the plant that I'm experiencing water limitation, I'm going to not grow as much shoot, I'm going to invest more in roots. So we, and then, there's others that had bigger roots, but generally roots do respond, and even in our situation with the pots. So we followed that up with more detailed experimentation and showed that the panel, the plants on the two groups of plants on the left are well watered. One is Suevo and the other one is the NIL. But if you look at the ones that were water limited in tubes and so on, that the roots were clearly more robust in response to water stress in the NIL that we had identified. So we followed that up with a lysimeter experiment where we were looking at the canopy level in the pot still, but canopy level conductance of the NIL 20-2 and the elite parent. under both control and drought conditions. And we did this in over several days, and that's kind of shown in the top panel. And we did it every few hours, so every 2 hours. So what we found was quite interesting. We found that the IL, which is the NIL 20-2, it was showing a difference in how much water it was going through it very early in the early part of the day. So if you looked at afternoon, you would not observe a difference. So essentially what it was indicating to us is that when the temperatures are cooler and the radiation is lower still, which is shown on the other y-axis, the NILs take that opportunity and they let a lot of water go through and also fix carbon in the process. Just so that just to kind of remind you that typical plant would lose about 300 to 400 molecules of water to bring in 1 carbon dioxide molecule. And that's just the inherent nature of the size of these 2 molecules are. So plants have not got much going in terms of changing the size of carbon dioxide molecule or water molecule, so they lose about 90% of the water that they take up. So this was also kind of highlighted in the panels below under well water condition and limited water condition, where we have the vapor pressure deficit, which means like what's the demand for pulling the water through the plant at atmosphere level. So that's kind of gave us a very nice insight. But we also wanted to ask, what's the situation? This is all done in pots. How does this translate into, a field-like situation? So our collaborators found new collaborators who had better ideas about, how to do this in soil. So they used the electron resistivity tomography to compare the profiles of water at different depth for NIL 20-2 and the SWAVO parents. So essentially, it's 20-2 minus SWAVO. So if you look at the broken line, so if the line's kind of waving towards the left, that means that there's more resistivity in the for SWAVO. And if it goes towards the right, then there's more resistivity for the NIL. So essentially what it's saying that as you look at as the plant gets bigger, you get a clear, in underwater limited condition, a clear increase in the negative values, which would mean that the SWAVO has less water access. Other than at a certain depth, about 0.5 meters, you see that NILs are actually pulling a little bit more water from there, so there's less water around that situation, in that location. So that kind of tells us that, that's just one line that we picked and went in great detail to kind of find what the role that NIL might have in terms of adding plasticity for roots in response to water stress. So just to summarize, imaging is like an ultra powerful tool for physiology. It may not be super intuitive in the beginning because a picture's a picture, like you take pictures all the time, but pictures also can pick up very, very small differences precisely, and you can quantify it, which means that you can connect it to genetics and then start linking genes that you may have brought in from for these particular trait. So we also show that Wild Emmer, can bring a lot of powerful alleles for adaptation, improving the quality of the produce, the amount of the produce. So this would be one instance where we were particularly looking at drought adaptation. So similar things can be done with the same material. Finally, I just didn't have much time for this, but we are exploring to see exactly what genes were brought in with this NIL, so that we can start precisely making markers for breeding and other efforts for that. With that, I would like to acknowledge the UNL team that was involved in, and Dr. Wada was part of that team, Hongfeng Yu and his group and my lab members, and our collaborators in Zvi Peleg's group did all the breeding and the genetics, and then Dr. Schwartz was involved in the soil work. And thank you for funding from these sources, and thank you for your attention.
Thank you so much. I don't have my phone, I don't have my watch. I forgot it home, so I don't know how much time we have for questions, but you'll tell us. I have a quick question to start. It's more philosophical. As you are presenting, it occurred to me. So we know climate is changing very rapidly. Losses are happening in crops. In fact, you told me with every degree increase in temperature, we're observing 6% decline in wheat production, correct?
Yeah. In fact, if you looked at county level data for in state of Nebraska, the genotypes or the hybrids that farmers are growing, they're not seeing as much of a genetic gain in parts where the temperatures kind of get higher, especially the nighttime temperatures. So yeah, so there's definitely
that's only an introduction to my question. At the same time, if we continue our business as usual production and consumption habits, it's projected that we're going to need by 2050 1.6 Earth. That means 60% more Earth that we don't have currently. So how optimistic are you that we can sustainably meet our food, fiber, fuel production by 2050 with all these constraints that we know of? I don't know. That's why it's a more of a philosophical question.
I think it's just the fact that I had to take a deep breath, and I saw some people kind of start stretching a little bit. Maybe it's towards the end of the conference, but I think it does beg to be asked, right? And so I think the genetic sources of adaptation are there, because we have fairly extreme environments on this planet. Have the very cold places, very dry places, very hot places, very flooded places. So we have increasingly more and more the tools that we need. I think we have the genetic resources because of, those things are adapted to those environments. It's just a matter of how much effort and how much new ideas to bring things that already exist, and how do we overcome that? Like, how do we diversify our food? While it feels good at the beginning of the talk to say that, I work on rice and wheat, and I'm sure many of my colleagues feel similarly, they're very important, but it also is kind of scary that you would depend, like the whole species, our species depends on just a handful of species for our, largely our survival. We don't even go past 30 species or crops to actually get 98% of everything that we eat. So yeah, so there's definitely, those are also important questions to ask, like, how diversified can our diet be? How much, could we bring more tolerance to water and temperature through that? Yeah, like by not only changing that particular species, but actually adding more species.
Yeah. Other questions?
Yes.
Into the modern cultivars, so that we can kind of marry this. So that we can marry gene discovery with modern cultivars, so that we can, more optimistically beef up our modern cultivars with these really, really unique traits that you're discovering in land races.
Kamal, do you want to repeat the question for the rest?
Yes. So the question is, we discover the genetics and the genes, and how does gene editing or biotechnology come into picture? I think that may be the game in town, right? Because I think the gene editing is very, very powerful. My laboratory uses it for rice very easily. And so I think it is already, I was in a conference last month in India, and it's already there. Like they're using not just, genetic background for editing to demonstrate the function of the gene. They're actually putting it in mustard plants, in elite cultivars that farmers are using to decrease the toxin content. So there's definitely a shift that I observed there that was quite striking for me. And so I think gene editing or one or the other form of it, as we enhance that technology, is we do have the tools now. Right, there's basically just a need for effort and some regulations that we need to revisit, and also the cost of how you overcome those regulations, and access to materials that would really get us closer to editing the lines that the farmers are going to grow. It's not like 15 years behind, right? Like something that they're growing, we can actually take and edit that. So there's some hurdles there, technology-wise, but I think it's not like a thing that you would say, okay, let's forget about this, find another way. No, I think you're right. The gene editing, or whatever version it's called in the next decade or so, is the way to go at this point.
And I would add to our friends in the EU and other countries, the policy and regulations in other countries, sometimes they're impeding and putting a barrier towards progress because of some of these regulations for, let's say, GMOs, and in some countries even gene editing.
Yeah, I think the gene editing is not going to leave an extra gene behind. And every time you go out in the sun, or the plant's growing in the sun, there's DNA being broken all the time. It's not fixed 100% correctly. So you are accumulating mutations. So we're at a point where we can change 1 or 2 base pairs here or there out of, let's say, rice would be, 370 million base pairs. So 1 or 2 base pairs here and there. Our cells are making that kind of mistake every time they replicate to grow. So is the plant cell. So it's not, obviously this should not be a blanket on that, but the idea is like we take a lot more risk getting in our cars than actually having a grain of wheat or a loaf of bread with one small change in the DNA.
We have a question here.
Thank you for your presentation. I found it very interesting. So
okay.
I'm here.
Okay.
I'd like to throw in a little bit of an Australian perspective here. So for the last 100 years, much of the breeding has gone on initially to provide resistance to viruses, the rusts, but more recently it's all about increasing production. But with increased production, what we've found is that we now have a considerable lack of micronutrients in the final product in the grains. And I'm just wondering, is that something that you have looked at or considered? And I'm wondering whether the option of going back to the older cultivars are maybe a way of trying to return the nutrition to the products that we're now producing.
Yeah, so that's a very, very good question. So the emphasis for a long time has been on the weight of the grain, so hence the breeding and the farming practices. But the other aspect of my lab's work is on temperature, and we've studied the impact of temperature on grain development quite extensively for the last 7-8 years. And what we've found is that you could have, if you heat stress a developing grain, it's going to change something such as it may have slightly lower starch and maybe a little bit more protein. But we've also found is that the weight of the grain, if you have a kilo or a pound, depending on what units you like, of rice grain, like a bowl, same weight, but if once treated with was exposed to high temperatures, you could have a lot less of that starch available. The way it is packaged, it's not digestible. So essentially what I'm telling you is that you could pay $10 a pound for the grain, and another, in a few months, pay for another $10 a pound for a grain. You may not get the same calories. And the profile changes not only in terms of the amount of calories, but going back to your nutrition, what we found is that the starch chains becomes much smaller. So you would get a lot more energy or glucose right away. So that would increase your glycemic index, but it's actually not very good because you would ideally want your meal to get digested over time. So there's definitely, an impact that we know for sure for temperature, but we are hoping to explore that from the water side as well. But yes, so the nutritional side does change, but there are also cultivars that we found that don't do that. So the question is, if we can figure out why they don't do it, then that's kind of a nice target for us, because with these advanced genome sequences that we have for these 3 species that feed us, we should be able to precisely pinpoint the gene, and even within the gene, what we need to change to improve the nutritional quality. But going back to finding it in the wild lilies, that definitely, is an option too, especially for iron and zinc in wheat. People have explored that quite extensively.
And I want to add, in the natural systems in Kansas, where they had severe drought in the past few years, what they have found is that the nutritional value of those forages in natural grasslands have changed. In some cases, increase in toxins that make them less palatable for animals. So that's something we aren't even thinking much about moving forward. So very good question. Thank you. Other questions from the audience? Yes, Dinos.
What's the role of AI in these kinds of research?
The immediate role of
Do you want to repeat the question?
what is the role? This question is, what's the role of AI in this? So the immediate role is that we have layers and layers of data, and we are increasingly generating more of that without capturing all that information in a context from which we can get increased options, right? So we go through phenomics, we have information from satellites, from drones, but using all of that information rather than making a clear linear line saying, okay, this is how I'm going to use this information across these layers. I think that's how human brain works, but we basically need to unhumanize. I don't want to say dehumanize. I think that would not be relevant. Unhumanize how we learn and extract actionable information from this data. So I think that's just the dimensionality of it itself would beg for a tool like AI. And I'm sure this is a very, very limited immediate answer, but yeah, there's a lot more that we can learn and use information from. But I do feel that my program and programs across the country and the world, we waste a lot of data. We pride ourselves in generating more and more data on the basis of technology, but we're also wasting a lot of data. It's just going through the sides and underneath and over us. So yeah.
All right. Any other questions? Okay. Well, thank you. Harkamal's going to be here for lunch. And if you have more things to discuss with him, please feel free. And I want to pitch something. After lunch, I'm taking a couple of colleagues across the street to see the greenhouse. So if you're interested, let me know, and I'll take you as well with me. I don't know if tomorrow it's part of the field trip as well, Peter. This afternoon it is? Okay, then. Well, join me thanking Harkamal and preparing for our final.
Thank you.
Okay, Peter. It is that time. Thank you. So, Peter doesn't need any introduction. He has been our fearless leader for several years now. And we've kind of asked him to give the final note and his final reflections on being a director.
Wow, you're teeing that up.
Thank you. Well, thank you. Please join me welcoming Peter.
Thank you. Thank you, Tala. Thank you, Harkamal. And maybe going back to the first evening, Harkamal Walia is also the Human Chair of between agronomy and the Doherty Water for Food Institute. So it's quite fitting to sort of end the session bringing those kind of alignments together. And always very interesting to see around the different aspects of what we're trying to achieve in securing water for food, and certainly the crops and the crop breeding and then the crop management are hugely significant in this very changing world. With this, I'm supposed to have a video up, a quick video to run, just kind of recapping the 15 years a little bit. You'll recognize some faces in it. I don't know, I assume it's ready to go.
Over the past 15 years, DWFI has made a profound impact by bridging the gap between research and real-world application, fostering partnerships across sectors, and equipping policymakers, farmers, and communities with the knowledge and tools needed to use water more efficiently.
What makes DWFI vital and unique is our role in bridging two often separate worlds. We bring agriculture into the water discussions and water into the agricultural policy making.
It could be anywhere in the world, but it is uniquely fitting that it's in Nebraska. We sit on top of the Ogallala Aquifer, and the University of Nebraska system have been dealing with water for food for more than a century. J.B. Milligan, who was president of the University of Nebraska at the time, 15 years ago, 17 years ago, we were talking about the university's priorities, and one of the top priorities was water.
The work being done isn't just theoretical. It's transforming the way farmers manage water, helping policymakers make better decisions, and improving food security for millions of people worldwide.
Doherty Water for Food is truly impactful globally. I'm most proud of JB and I giving birth to this institute and having the opportunity to present this idea to Bob Doherty before he passed away. So his initial commitment of $50 million, if you will, as seed money, but important seed money for this institute, is probably what I'm the most proud of.
Seeing the impact of our research, the strength of our partnerships, and the achievements of our students and scholars has been deeply inspiring. But above all, I'm proud that the DWFI is making a tangible difference, not just here in Nebraska, across the nation, but truly around the globe.
DWFI's impact over the past 15 years has been driven by the dedication of the people and partnerships behind it. None of this would have been possible without your support. Thank you for being an essential part of the journey and the future ahead.
Thank you. I'm always very uncomfortable with watching myself on screen, but the things I have to do. What I would like to stress out of that is, and it's the timeline. 15 years sounds like a long time, But actually, if you look at the water and food security journey, and we've been hearing some of this, the 150-year journey in the Nile and the progress here for the last 150 years. And in other parts of the world, it goes back 2,000 years. So it's a long journey. We develop our efforts on the shoulders of giants as we try to address these issues. But I think most importantly, and we've been reminded of this week, but also not just in this conference, working with some other stakeholders here in the state, is the power of partnership. And that really what it comes down to. It's the partners. You saw Moens Bai, who was here, who is he and the president of the university, plus other leaders in the state, really helped set up this institute. But it's then all the different, my team, the team that works in the institute, this team that works across the university, how we partner outside through our networks. And so it's important that many of us have experience in other contexts, but also work in Nebraska as well and tie all these threads together. But in the end, any kind of celebration around impacts and outcomes in this space, as we realize all the complexities, comes down to the partners. We cannot do this. We cannot even create impact in Nebraska without the many partners we have here in the state. So it really belongs to you all, but also your colleagues and your network of partners, because this, I feel the mission of the Water for Food Institute is the mission of many of us in this room. So thank you for your dedication, for your collaborative spirit, and for your energy in surviving these 2 or 3 days of many sessions. One of the feedback I received was just how difficult it was to get into the best session, the breakout session they wanted to be in. It's been quite, and I also was the same. This morning I wanted to be in all 3, and yesterday I wanted to be in all 3. The videos it's all been recorded, will be edited, but it'll be up within a month. All the sessions will be up within a month. So you can learn all the aspects of the things we've been trying to cover here this week. I think starting off with the first day, I'll come back to the first session, but certainly the Heuermann lecture, really, and I sort of tongue-in-cheek said this at the end in terms of it's more nutrition per drop. But it's really trying to think about the conversation that it's not just productivity, it's how do we bring in the livestock component of it, the other crops we produce, and higher value crops from a nutritional perspective. But also hearing the producer's perspective on this, how difficult it is to do that. Here we run this that we can quickly shift from corn to tomatoes. And then you heard from our friends in Schooler talking about what it takes to even introduce canola into the crop breeding cycle here because of the infrastructure and the connections to markets and all the aspects around that, the insurance. So all the signals that are sent from the policy level. Again, that piece of having the farmers here and having producers from El Salvador and from Kenya and from 2 or 3 different parts of Nebraska and Brazil. This lets us hear about just the complexities they're dealing with, but also the kind of fighting spirit they have in terms of trying to get agriculture to work in these areas. Maybe a reminder of all of that, something I find myself having to stress is those farmers they've got so many variables they're dealing with, no matter what scale they're at. And water, to us, seems super important. It is, but it's actually, they want to manage that piece of it out. And so it ends up being often, and actually Claudia raised this in the Heuermann lecture, was the small farmers, and particularly the women farmers. The most precious commodity to them in implementing an agricultural practice was actually their time. It's not their water, it's maybe not even their energy, but often it's quite different. But if you go out to these large-scale farmers here, in the middle of the irrigation season, they're not worrying about how much water they're using, unless it's automatic. They're worrying about their time, and then energy becomes the cost of energy becomes a factor and so forth. But if you've also worked with producers here, you realize the big unknown in this part of the world is hail. And that can really wreck your day as a farmer. I think and this is one of the things I encourage you to actually go back and look at some of these sessions, because some of the interconnected challenges, you'll see very common themes through some of these sessions. Again, that can be viewed as complexity, but it's also understanding. If you pull one thread here, there's something else that moves over in another space. And again, when it comes to drought, health, nutrition, and then how we educate, capacity building, the session yesterday on capacity building and development and education, a hugely important aspect we're hearing from all our partners is how do we better educate, and this is something here at the university we're working on, but also working with partners to help address those issues. Again, I'm coming back to thanking you all for your participation, your insights, and your energy. I think we're a little bit back on numbers this year, but the energy has been notably gone up a notch, and I think that's fantastic to see. I do hope. I was tasked with addressing outcomes. I think this is a journey. I've got my thoughts on outcomes. Likely, and this has happened with previous conferences, some of these synergies and discussions go on and actually create some really spectacular outcomes, and I encourage you to do that, and we look forward to hearing your stories next time, and we look forward to the ideas and partnerships and how we all can connect and how we can help facilitate those partnerships. With that, let me turn to the part. We should now have a PowerPoint with slides, so can I have that up, please? So now I just want to quickly say one more thank you. Again, I will thank you all. This is really fabulous to see so many of you still here, engaged and participating. Very appreciative to Valmont, who are our platinum sponsors, and then other sponsors of the conference. A conference like this takes a lot of, it certainly takes a village, of partners to put on something like this. That really requires significant logistics. And our partners, these sponsors share our mission and different aspects of our mission on water and food security and help us to keep registration fees manageable. Thank you also to the exhibitors. I hope you managed to visit the exhibition space. Hopefully you had the opportunity to talk to them all and pick up cards, and I suppose it's now QR codes and connections for the future. I would like to recognize our board of directors. Moens was here, Jeff Gold was on video at least, Felicia was here and very active, and the others have been basically participating in different ways as we go forward. And then we also have an international advisory panel, and we met on, that seems so long ago now, it's Monday morning. So we met, and again, we bring most of our international panel members in at that time. So Uma couldn't join us, but she joined us virtually. Rachel McDonald, the same, couldn't. But then Bob Meany, who spoke yesterday, Letitia Obeng could not join us, but she was very active online. A great supporter of the Institute. Martin Passman just left, and Rorik Paulman, one of the producers who's very active in our conference and very much involved in our program. And then it would help if I read the one in front of me, wouldn't it? Big thank you to the staff and the team, and that team. When I say we, it extends out beyond the university and into many of our partner organizations. But certainly the staff here, Francis, our director, had to step out. We're dealing with a small urgent item that we have to work on, but certainly she's been the main kind of guiding hand in putting this together. We're very far ahead with developing our program this year. We've also learned lessons of what we'll do different next time. But also Amber, who has dealt with a lot of the logistics and every element of this, and it's fascinating with the different delegations that come from overseas and all the complexities they have about getting here and arriving on time, and all kinds of things go on in the background. And I must admit, this one, there was very few crises bubbling to the surface. The program committee and Tala and others have been very involved in that, and a lot of work went into that, and that came through in the session. So again, thank you all for doing that. The volunteers, we've pulled in students and others to help volunteer on this. And then the Visit Lincoln volunteers, so our colleagues and partners here in the city join to help us put on an event like this. Next slide. The vendors, I think everyone thought Country Pines was I had never been there, that venue last night, but yeah, that definitely hit a high point. We have the software, the different communications people, our media and communications colleagues across the university, even the suppliers of beer and Wren's Display and Sunburst. The Scarlet Hotel, you know, I'm sure that our meager accommodation was suited for you, because it is a wonderful hotel, although I've never actually stayed there. UNL Alumni Association, the police, the university police. It gets complicated because we sit on a private site, and so even the policing here is the city, is it the university. So this is, and always making sure that we stay safe. And then venue catering, and I would say the food was very good. I thought it was very good this time, so I'm pleased to have been getting positive feedback on that. Next slide, please. Well, with that, this is the end of our time here. I won't keep you. The resilient future, I think you need your lunch to get through that resilient future. Again, thank you all for coming. You're welcome to come back here. Don't be shy. Please reach out to us, and we will also try to connect with the different folks that are keen on moving things forward. Thank you all.